So today, I'm going to try and cover, in the time that we have, I'm going to give a sort of whirlwind tour of biological thermodynamics. And apologies if you're a physicist or you've studied this at great depth at graduate level. This is a sort of very basic and simple introduction to thermodynamics. The idea of this is just to get people to think about how thermodynamics are underlying a lot of processes that we're interested in. And there are a number of interesting aspects of thermodynamics that can kind of like influence either methodology we might want to use or behavior of our systems. I'm then going to talk about biological calorimetry, which is what we're here to understand, but in a more general context about why we would want to do it and how we might think about doing it. And then finally, talk about one of the most popular forms of calorimetry for biological applications, ITC, isothermal titration calorimetry. If I go over the time, I apologize, but I will try and keep it under an hour. So certainly in this building, this is the kind of framework of what we're interested in. Obviously, we're not only looking at proteins, but in this context, we could think about proteins doing a whole variety of things that we're interested in, forming complexes, inserting into membranes, interacting with nucleic acids or ligands, and maybe multimerizing. And there are probably still people doing crystallography, for example, but not so many. Cryo-EM would kind of come off here at the big complex. And also we can think about proteins doing things that we might be interested in, like forming ordered associates and sort of some sort of fibrillar structure, and also proteins doing things that we don't want them to do, like misfold or aggregate and behave horribly. So all of this is driven by consolidated, meaning kind of like they're acting together, individually weak non-covalent interactions. And this means that many of these processes are reversible. So we can go from unfolded to folded and back and so on. So if we just think about protein folding or protein ligand binding as simple examples of biological equilibria that we might be studying, at the equilibrium, by this we mean the endpoint or the steady state levels of the reactants, if we want to think about them as that. There's no net change in the concentration of either side of this equilibrium. And therefore we can define the reaction position, which is then kind of like the biological effect that we're interested in, as simply the ratio of the concentration of the reacting components, if you like, in this case, unfolded and folded protein, or protein and ligand and the complex. So the equilibrium constant can be defined by the ratio of these concentrations, and it's also equal to the ratio of the forward and the reverse rates for these reactions. So this equilibrium constant can be thought about in a kind of logarithmic sense, makes it easier to talk about. And we introduce a couple of constants, the gas constant and temperature. Obviously, temperature is constant for isothermal work, but not constant otherwise. So the Gibbs free energy, this delta G, is just a logarithmic representation of that equilibrium position. And the Gibbs free energy has components of this thing, enthalpy, and this other thing called entropy in this second equation. And where this Gibbs energy is negative, the log equilibrium, so the equilibrium constant and its log must have been a positive number. So that means that the equilibrium must be greater than 1, and therefore the products are in excess. So negative delta G means that we drive the reaction towards, I'm calling them products, but in this case it's complex. And a useful kind of back of an envelope kind of calculation is that if you change the equilibrium constant by an order of magnitude. This gives a change in the log of 2.303. And at 25 degrees C, RT is equal to 0.59 kilocalories per mole. This means that for an order of magnitude change in equilibrium constant, we need a change in free energy of 1.36 kcals per mole. I should apologize at this point in that everything I refer to will be in calories, which is not the SI unit. Joules is the SI unit, which is 4.184 joules per calorie. But obviously it's calorimetry, so we're sort of married to that particular unit. So what are enthalpy and entropy? This is a very sort of simplified view. You can think of systems as naturally progressing to a lower internal energy level, and under constant pressure. This corresponds to a lower enthalpy. We don't see things sort of stable in a very energetic state. Things tend to progress to a lower energy level. And this change in enthalpy is negative when we progress to a lower energy level, and heat is released. And because this change in enthalpy is negative, we're driving the equilibrium towards the product. So negative delta H gives us more product. However, systems will want more ways of configuring with whatever energy level we achieve. And this is just simply a statistical phenomenon. If you think about anything, the more ways there are of obtaining a particular outcome, the more likely is that outcome. That means that the more, if we call it disorder, will increase the probability of forming the products. So when delta S is positive, that means there are more ways of configuring with this energy level that we've achieved. Then that means that because of the negative T in this equation here, that means that we're going to generate more negative Gibbs energy and favor products. Okay, so where does the equilibrium actually end up? Well, it's just simply, because of the equation, a balance between this enthalpy and then the entropy multiplied by the temperature. That doesn't mean that the reaction won't proceed or the complex won't form if one of the components is unfavorable. Here, both entropy or the entropy component and the enthalpy are favorable, but here we have unfavorable enthalpy, here we have unfavorable entropy. But nevertheless, because the other component is larger, we're still going to progress towards product formation, or in this case, the complex. So these terms of, or these parameters, enthalpy and entropy, are they actually fixed? Well, no, they're not, because particularly in biological processes, there are obviously changes in non-covalent interactions. There may be changes in conformation, and associated with all of this, there are going to be large changes of solvation of the system. And all of these things affect the heat capacity of our reacting components. Heat capacity is just the energy that you need to put into the system to increase its temperature by 1 degree. So obviously the units are calories per mole per degree. Biological reactions mostly all have large changes in heat capacity. And heat capacity is important because the integral of the heat capacity function, or the integral of the heat capacity divided by temperature function, from 0 Kelvin up to the temperature that you're working at, represents the enthalpy and entropy levels for the system. And therefore, because of the significant change in delta Cp, both delta H and delta S become very temperature dependent, and also therefore delta G, since it's a product of the difference of these two parameters. And because of this relation here, where we have this logarithmic term, this means that the entropy term is nonlinear, and that generates nonlinearity in the free energy as well. And that has some interesting consequences and that we can observe. So we have this marked curvature in the free energy function. As you can see here, I've just simulated some data with a protein with a melting temperature of 320 Kelvin and an enthalpy of 50 kilocalories per mole. So here we can see the free energy isn't just a straight line, it's this parabolic function, and it has a maxima here at different temperatures. And that maxima occurs when the entropy is equal to 0, because the derivative of the free energy as a function of temperature is equal to the entropy. Below this maxima, there's a temperature, there's a zone of temperature where destabilization occurs. So the free energy is going down, and potentially here, the simulations predict that the protein would unfold again if we cooled it down far enough. This is known as cold denaturation, cold unfolding temperature. And this is in addition to the conventional melting temperature that we would think about when we do thermal melts. The larger the delta Cp and the smaller is the enthalpy, then the more curvature we generate and the higher this cold unfolding temperature becomes. You can see here these three different simulations. The blue one has a heat capacity change of 1 kcal per mole. The green one is 1.3 and the red is 1.6. So this is with the same enthalpy of 50 kcal per mole. So the larger is delta Cp, the more curved it becomes. Why is that interesting? Well, first of all, this is a sort of an interesting physical prediction and has been observed experimentally under a number of occasions. But one thing to think about is the interfaces in biomolecular complexes can often be low enthalpy, so they may not be stabilized by very many interactions, but they may have a relatively large delta Cp because there's either changes in conformation when we form the complex, or there's exclusion of solvents from the interface in the complex. And so cold inactivation of many multimeric enzymes at 4 degrees C can occur via subunit dissociation. So basically the complex just falls apart when you cool it down, and it's all to do with the fact that this free energy is decreasing and stability is going down. This has been documented for many glycolytic enzymes, many of which are multimeric. So just to make you think, sometimes, keeping everything on ice and being paranoid about that may not actually ultimately be the optimal solution. Another thing to think about with all this curvature in delta C, which is induced by delta Cp in the free energy function, is think about how organisms might adapt to become thermophilic. So if we look at an organism here with our parameter and Tm as a protein here at 320 Kelvin, if we just shift everything up, so we keep all the parameters the same, and we make the protein unfold at 370 Kelvin, then we bring this cold denaturation temperature right up here. So this organism wouldn't be able to readapt or live at lower temperatures. But by changing parameters in the protein's thermodynamics, for example, lowering the delta Cp would generate the red function. So then we maintain stability over a much wider range, and yet can still have this higher melting temperature. And then equally also here, you can see if you increase the enthalpy, you can generate the same effect. But in this case, you may generate stability, which is too large. Obviously, proteins need to be turned over. And so it's not a good idea to have sort of unlimited amounts of free energy. So, okay, so we've thought about how the enthalpy and the entropy affect the equilibrium position from the equations, and we thought about how delta Cp affects it and combined with temperature. So can anything else affect the equilibrium position, which is kind of what we're interested in thinking about the biology? Well, yes, actually pretty well everything can affect the equilibrium position. And this all comes about because of this law of mass action, or Le Chatelier's principle. And he stated it as when a system at dynamic equilibrium is disturbed, the equilibrium position will shift in the direction which tends to minimize or counteract the effect of the disturbance. So that's all of this sort of wordy. But here's a simple example of mass action or in action. So if we think about these two proteins interacting here, the pKa's of groups, ionizable groups, will change on complex formation due to a different environment, or they may be involved in specific electrostatic interactions. So their pKa shift. Thus, when the equilibrium position changes, when we go from this to this, we're going to change the proton concentration. Although obviously in our experiment, we will buffer the pH using a biological buffer, as it is in biology as well. But because of this fact that the equilibrium changes the proton concentration, the mass action would therefore tell us that the proton concentration will affect the equilibrium position. And this is how you get pH dependence of free energy. Okay, so free energy, if there's changes in protonation, that means that pH is going to then modulate our stability in free energy. Here's some more examples. You can think about an oligomer, the equilibrium position between this folded tetramer and unfolded monomers. The equilibrium position changes the concentration of molecules. Therefore, by mass action, concentration of molecules will affect the equilibrium position. This means that the higher the concentration, the more we have of the folded species, the higher will be its melting temperature. Similarly, here's another example. The equilibrium, here we're thinking about a ligand binding to a protein. The equilibrium involves changes in the concentration of the free ligand. So we've only got 2 here, and over here we've got 6. Therefore, the equilibrium stability that could be measured by the chemical stability or the melting temperature will depend on the concentration of the ligands. And this is the basis of thermal shift assays that we're going to hear about in the next talk on Tuesday. So biological calorimetry, why would we do it and how would we do it? Well, calorimetry by definition means basically to measure heat. And the heat of the process under constant pressure is effectively equal to the change in the enthalpy of the process. If we can measure this enthalpy or the heat during a reaction as a function of the equilibrium position in some sort of a titration, then we can determine the equilibrium constant and we can therefore get the free energy. Once we've measured the enthalpy and the free energy, we can obviously then obtain the entropy. And this gives us a complete thermodynamic description of the equilibrium process we're studying. So this in itself seems like a pretty good reason to think about doing calorimetry, particularly on biological systems. However, in addition to that, pretty well all biological processes will have an associated enthalpy, or all reactions or conformation or sort of equilibrium processes that we might want to study will have an associated enthalpy. The enthalpy is our signal. So everything's going to have a signal. We can measure anything. Conformational change, melting, binding, interactions, enzyme turnover, protein turnover, enzyme catalysis. You think, pretty well anything can be measured. Calorimetry is also, by definition, a non-optical and a label-free method. We're just looking at the molecules. We're measuring the heat that they generate. Either they release it or they take it up. And so therefore, we don't need any specific groups or labels on our system. It can also use optically difficult conditions to work under, crude cell lysate, turbid suspensions, unusual solvents, high backgrounds of other molecules, and we can apply it to many, many systems at the molecular up to a cellular level, and even the whole organism level, if you think about bacteria, etc. As long as you can get them in the calorimeter, then we can measure. And for this reason, because it's There is no requirement for specific routes and the universal signal associated with all processes. This means that we don't really need any method or assay development. So potentially it's a very rapid method that you might use in your pipeline. So not surprisingly, calorimetry was actually one of the earliest techniques ever reported in the literature. And this comes, very early experiments were done by Lavoisier using this ice calorimeter, in which he showed that respiration was effectively oxidation of carbon or combustion. The way he did this was to put something in this inner chamber. They would measure the amount of carbon dioxide given off during whatever they were doing, say burning charcoal or putting this animal into the calorimeter. For some reason, they used the guinea pig. I don't know why. And then the ice around the calorimeter would melt as a result of the heat that was produced within the calorimeter. They would collect that, and then knowing the thermodynamics of the solid to liquid ice transition, they could calculate how much energy had been released. The other layers are kind of insulation. So there are lots of other types of calorimeter used in many, many different areas. You have combustion calorimetry due to associated with fire safety, so on. How much calorie does your, what's the heat evolved when you burn your sofa or an office or something? This is part of the Large Hadron Collider. One of the experiments there was this Atlas experiment, which is effectively a calorimeter. Is a biological calorimeter. You have sort of human calorimeters for nutritional studies and so on, and simple calorimeters that you might have come across in school, where you sort of burn a peanut and look at the energy of that. So in terms of biocalorimetry, we think about the heat, the enthalpy that we might get for a typical protein-protein interaction. It's going to be maybe, if we're lucky, say 10 kcals per mole. If the KD is 50 nanomole and the delta S is 0. So you'll get 10 kcal per mole if we have a KD of 50 nanomole with no entropic contribution. So if we have 1 mil of 50 micromolar protein solution, this is going to produce 5 times 10 to the minus 4 calories of heat when it binds to the partner. And this is going to melt about 6 micrograms of ice that we could collect in Lavoisier's ice calorimeter, or it would heat up 1 mil of water by 5 times 10 to the minus 4 of a degree. So that's going to be pretty difficult to measure if we approach the biological calorimetry problem as direct heat transfer calorimetry. So the way around that is to use power compensation biocalorimeters. And in this case, we have 2 cells. We have a reference cell, which is almost as identical as it can be to our sample cell. And we measure the difference in temperature between these 2 cells using a very precise thermopile. And we try to keep this difference in temperature as constant as we can using a series of feedback heaters. And the additional differential or difference power that we put into the sample cell when something happens, either the protein unfolds because we're heating up the whole system, or we get energy or enthalpy released or taken up by the system because we titrate in sample and the molecules interact. This difference power is effectively equal to the measured excess heat, which is the enthalpy. So this is kind of what they look like. This is an isothermal titration calorimeter. Has a syringe on the side, and we can do titrations. And we work at a fixed temperature between 4 and 70 degrees C. And we can determine equilibrium constants between millimole and down to nanomole levels. We also have differential scanning calorimetry, and in this we look at temperature-induced transitions, so melting. We can increase or decrease the temperature, and we have a range of 0 up to 130 centigrade because we put a small amount excess pressure on top of the cell to stop the liquids boiling. So here's the kind of mistake that a lot of people make, including me. When you come into this field of calorimetry, you're kind of thinking, well, I can measure thermodynamics, and these are the fundamental properties that are driving the position of the equilibrium, is telling me what the reaction is going to do. And then with protein engineering, I can fiddle around with the protein, add in extra interactions, make the thing bind tighter, discover a drug, become rich, become famous, get lots of papers, etc. That's all very well, but the problem that we have is that the measured heat, the enthalpy that we're measuring in our calorimeter, is a global and non-specific probe, and what calorimetry measures is all of the heat that's released in the process. So that's the specific structural interactions that we can see when we look at a structure in a paper on a page, but it's also lots of changes in solvation, shifts in pKa's, and changes in protonation, changes in dynamics of the system, etc., etc. So calorimetry is really highlighting the fact that these additional changes are also part of the equilibrium. Everything that's happening is going to contribute to our observed and measured macroscopic enthalpy. There's also a phenomena in terms of manipulating interactions of enthalpy-entropy compensation that tends to try confound rational structure-based thermodynamic design. And that's basically means that if you increase the enthalpy by introducing interactions, that increase in enthalpy to the free energy is generally very well compensated for by a decrease in entropy of the system. So what you gain in enthalpy, you tend to lose in entropy, and vice versa. So just to re-emphasize the fact, what we're talking about is when these molecules come together in terms of interacting in solution and solvation, we have water molecules that are associated with this potential interface here, and they're quite different from water molecules that are in bulk solution. So they're likely to be more immobilized, particularly if we have hydrophobic surfaces, etc. So when they come together, these water molecules are often displaced out back into the bulk solvent. So what we measure, our observed enthalpy, is the enthalpy of interaction between A and B, if you like, thinking about interacting in vacuo, but also an enthalpy resulting from this solvation of these molecules going out into bulk. And obviously I've drawn this just showing here, but obviously the whole surface of these molecules is hydrated, and effects can be propagated over longer distances. And there are different shells of water associated with proteins. We can also add the element of ionization. And in this case, we often make measurements in buffer. And these buffers will have an associated enthalpy for ionization of the buffer. And therefore, if we have changes in pKa during our interaction, then the protons that are liberated or taken up by the system during the interaction will have an associated enthalpy of ionization. And so therefore, the measured enthalpy is starting to be the product of many, many different contributions. This is why it's extremely difficult to make statements about what the numerical, the value of enthalpy means in an isolated situation. One thing that this does give us, though, is that calorimetry can be used to measure very directly the net protonation changes during some interaction that we're interested in. This is because if we perform the measurements in buffer, there's an associated enthalpy of ionization of buffers, and these here, for example, are well tabulated. This means that we have a measured enthalpy, which is our enthalpy of interaction, some solvation contribution, then the change in protonation, the effective change in protonation, times the enthalpy of ionization. So if we make measurements in a series of different buffers at the same pH, and with the same amount of salt. We plot these out as a function of the ionization enthalpy of the buffer that we're using. Then the intercept is where we have an absence of contribution from this ionization effect. And the slope of that plot was directly equal to the number of protons involved in our system. This also raises another point that many people don't think about or aren't aware of, and it's back to mass action again. So buffer ionization produces heat, which we can think about as enthalpy. It's part of our signal when we measure it isothermally, for example. And this would, the ionization produces heat, and this would change the temperature of the system. Therefore, through mass action again, the temperature changes ionization of our buffer equilibrium. So this means that buffer pH can be quite strongly dependent on temperature, with the largest changes in buffer pH depending on the value of the ionization enthalpy. In this column, you can see the change in pH per 1 degree of change in temperature. And you can see that phosphate, pKa 7.2, that's a pretty low enthalpy of ionization, and therefore a small change in pH as a function of temperature. So it's quite good for if you're wanting to do experiments where you're changing temperature a lot. In contrast, tris buffer has a very large enthalpy of ionization and therefore a correspondingly large change in pH as a function of temperature. And just to show that at pH, if we have tris buffer pH 8 at 25 degrees C, it will be 8.6 at 4 degrees C and 7.7 if we worked at 37. So again, thinking about thermodynamics and mass action and things helps us even to do very much more practical work in the lab, like choosing an appropriate buffer. So moving on to practical biocalorimetry. These are the calorimeters that we have here in the LMB. We've already seen that we have ITC with a titration option, and then DSC for making measurements as a function of or looking at temperature-induced melting. So ITC is generally the most popular and most commonly used technique. And a typical titration experiment, we have here instruments made or supplied by a company, Malvern Analytical, and they're called the ITC 200 series. We have both manual instruments and automated instruments. Then they have an active cell volume of 200 microliters inside the instrument. And the syringe here, which we can use to titrate in a ligand, has a volume of 40 microliters. So for a typical experiment, we would need 350 microliters of the target. That's the thing that we're going to put in the cell. And that should be at a concentration, an initial concentration anyway, between 10 and 50 micromolar of protein, of whatever we're using. When we load the instrument, we get about 75 microliter back. The cell is 200 microliter, but we need to fill the cell up to the top of this access tube here. So takes around about 275 microliter. The syringe is 40 microliter, but we need 70 microliter of ligand, and this should be on a range of at least 10 times that of what we have in the cell, so 100 to 500 micromolar. And in loading it, we get a small amount back as well. We're then going to make a series of sequential injections on the order of 2 or 3 microliters, maybe 15 to 20 injections. And although it's not shown here, within the instrument, there's an identical reference cell for this power compensation mode of action, and this is filled with solvent. So you can see already, probably a lot of people's jaws are sort of hitting the table there. ITC is a very sample-hungry technique. And so often it's used once evidence of binding from other techniques or the literature is well established. So it's not something that you might use as a primary tool if you were, for example, trying to find proteins that interact. You're going to end up burning through a lot of material. Because of this fact that calorimetry measures all the heat that's produced when we make the experiment or when we make the measurement, not only including these changes in solvation and changes in ionization, but when we actually physically inject solutions in, there may be additional heat. And so we're going to need to make a number of controls. And usually it's the heat of dilution of this component that's in the syringe, which is the main control that we need to take account of, and the largest source of additional signal. So here's some typical data. Here we have the experiment where we took 600 micromolar solution of a small peptide. It was injected into 45 micromolar, of protein solution. Series of injections, 2 microliters each in this case. And then here's the control measurement where we took just the peptide and injected that into buffer. So we can go through these data and integrate these individual peaks, and that gives us the amount of excess heat that occurred when we made these injections. And we can normalize that relative to the number of moles of material of the peptide that we injected in with each injection. You see, you end up with these curves here. This is the control, and this is the binding experiment, and we can correct the binding curve by subtracting the control or subtracting the linear function that we fit to the control, and then we can fit them. We can obtain a series of parameters, a stoichiometry value, in this case, equal to 1, a dissociation constant, and this was 1.7 micromolar. The enthalpy was minus 10.5 kcals per mole. And then from the equilibrium constant, we can get the free energy, therefore we can get the entropy out as well. So these plots with which ITC data are often displayed are sort of slightly unusual in the world of binding measurements. But it's interesting to think that they have, or to consider how there are actually different regions of these curves and how they're contributing to your robustness of your measurement in terms of constraining the fitting when you come to try and extract the values. So in this case, there's some more data that was obtained, and you can see these initial injections here are telling us a lot about the enthalpy or the amplitude, the total amount of heat that we're getting when we observe this binding. In this blue region here, we have a sort of sigmoidal inflection, and the slope of this sigmoidal inflection is telling us about the value of the KD. And the value of the molar ratio, which is what's plotted in these unusual ITC plots. This is the ratio of the how much ligand you've added divided by the concentration of the sample in the cell. The slope of this and the inflection point is telling us about the stoichiometry of the interaction. Then this data up here is telling us what the control heats are like, the end point of the reaction. And this shape of this curve, this sigmoidal curve, can be described by a unitless parameter, which in the field is known as the C value. And the C value is just equal to the concentration of target that you have in the cell divided by the KD for that interaction. So what it's doing is telling you how far you are above KD. For making an ITC measurement where you want to try and extract all of these parameters, the KD, the stoichiometry, and the enthalpy, then an optimal range for the C value is between 10 and 500. when you're trying to determine all of those parameters in one experiment. So the stoichiometry of the interaction, that's the number of molecules of what you're titrating in, binding to the target in the cell, is well determined when we have a C value of 10. So we're trying to look at where this inflection point is. You see the green data is C value of 10. But one thing about this determining the stoichiometry is that we're assuming that what we've done is to measure the concentration of the macromolecule in the calorimeter cell and the ligand in the syringe. We've measured them correctly, and both of these components are 100% pure and 100% native, I mean folded or active or binding competent. So although we need to measure the concentration accurately, we also assume that everything that we've measured is what we're trying to study, and that it's all happy and quite prepared to undergo the interaction. So the stoichiometry can also be viewed as an indicator of active binding site concentrations, meaning that if you get an N value, the stoichiometry less than 1, is telling you that that fraction, say it's 0.8, then it's telling you 20% of your material was dead and couldn't bind, participate in the interaction. Alternatively, you can also think about having this stoichiometry parameter during the fitting as just being a parameter that allows the measured concentrations to vary during the fitting, and therefore when you fit the data it looks great because the line goes through the points. So don't get too hung up on the stoichiometry value. The N and also other fitted values are particularly sensitive to errors in the concentration, particularly the component in the syringe. So when you have an error in the syringe concentration, you get an error in the stoichiometry, but also an error in the KD and the enthalpy. An error in the concentration of the material in the cell generally or only generates an error in the stoichiometry value. In practice, when N is equal to 1, plus minus 0.2, say, then we would say that is indicative of one-to-one binding. Other kind of numbers for N, like 1/2 or 2, may have some potential physical meaning, but often you're left with obtaining an N value that's a non-unit. Thinking about what's going on is also is what I'm trying to get across here, that it may well represent errors in your concentration or in your active fraction. I would also point out that calorimetry is not the only technique that can determine stoichiometry. The reason that people imagine it can determine stoichiometry is mainly because the concentration we need to work at often means that we're well above the KD of the interaction. And for that reason, stoichiometry becomes a fairly well constrained parameter during the fitting. So you can do any other type of optical assay, titration, binding experiment, and if you work under particular conditions where you're in excess of KD, if you think about the C value, when we have a C value of 10, we're 10 times above the KD with the components we're measuring. So what do we do if we want to study tight or weak binding, if we're going to be fixated on this C value between 10 and 500? So if we have a very low KD, very tight binding, that would mean that we're going to have to drop the protein concentration in the cell to try and maintain C below 500. The problem with that is we tend to run out of signal around about 5 micromolar of material put into the cell, and this corresponds to a KD around about 100 nanomole. However, I would point out that because of its non-optical nature, ITC is a perfect technique for doing displacement binding experiments, meaning that you would bind a weaker binding ligand and then displace that weaker binding ligand with your tighter binding ligand that you can't measure directly in one measurement. Using displacement methods, really super tight KDs, picomolar, have been determined and quantified quite accurately. So weak KDs would require increasing the protein concentration so that your C value doesn't become too small. And this would be beyond feasible limits, either for solubility or just being able to produce enough material. And so often these weaker binding experiments are done at low C values. In this case, the control heats of dilution are critical to correct the data. So if you're doing ITC, don't get too fixated on being exactly in the C value window. You can still get good data, but there may be, you may have to do additional controls. You may also have to do some constraining during the fitting. So far, we've just talked about using ITC to measure the dissociation constant or the equilibrium constant, dissociation constant, and the enthalpy and the stoichiometry. But it isn't only able to do that. So we've already talked about how if we, the value of delta Cp, the heat capacity change for an interaction, means that the enthalpy is strongly temperature dependent, because the enthalpy is the integral of heat capacity over a temperature range. So if we measure enthalpy over a series of different temperatures, then we can get the delta Cp for their interaction. This can be interpreted to some extent for a rigid body type interaction, and they typically have negative delta Cp's of binding. We can get the numerical value just by measuring the enthalpy, say in our ITC, at a series of different temperatures, and the slope of this plot is going to give us the delta Cp. If we have a non-constant value, we don't have a straight line here, or we have a very large value, or we have a positive value, this can indicate that you have some other types of coupled conformation involved in the experiment. This could be a conformational switch, or domain movement, or unfolding. I've mentioned that through mass action and through the use of different buffers, we can get at the enthalpy of ionization, or the protonation, we can get at the protonation change during a binding interaction. That's just an example there from the literature. We can use ITC to look at changes in conformation or molecularity, and this is often seen when you're looking at molecules, for example, coils, which are not often, when they're truncated out of their biological context, they're often not particularly stable. And quite commonly, when you look at the control heats, where you're just injecting the coil into buffer, you see a non-linear and decreasing endothermic type of titration like this. This is because the coil in the syringe is at sufficiently high concentration to be a dimer or a higher order species. But when we inject it, we inject 2 microliter into 200, so we drop the concentration by a factor of 100, and the coil falls apart. You can use this data to actually fit to get the KD for the coil, for the monomer dimer association, and I don't know any other technique where one could easily do that. It also means for routine ITC, you would tend to put the higher molecular component in the cell if that's possible. So sometimes when we've looked at protein DNA interactions, we've worked with very short pieces of double-stranded DNA, and therefore what we've done is put the DNA in the cell of the calorimeter, titrated in the protein from the syringe. Always do controls and reverse titrations. By that I mean doing the experiment in both directions, where you would have component A in the cell and titrate in B, and then do it with component B in the cell. Here's another example of something that can be extracted from ITC data. So we measure the the rate of heat production of the differential power during the injection. And when this heat production becomes slower than the response time of the instrument, then we are obtaining information about the kinetics of the process. When we make the injection and some heat is released or taken up within the cell, then the instrument is trying to re-establish this difference in temperature between the two cells at 0. And that electrical circuitry will have a fixed response function. When the kinetics of the heat release coming from the biology is slower than that process, then we start to see kinetics. And you commonly see this in a typical titration, that the injections near the end of the titration are broadened. And this is because the amount of free target in the cell is reduced as you continue to produce more and more of the complex. And there's software that will allow you to try and extract these kinetic parameters, which would be the K on and the K off, from the binding interaction. We haven't really applied this in great depth here, but certainly something to think about if you're interested in trying to maximize data from your experiments. And finally, just to say that ITC can also be applied to enzyme catalysis. And the big attraction of this is that it's a non-optical method, and you don't need to develop any particular assay that would give you some sort of colored product or some change in fluorescence or absorbance that you can measure. You can just go straight in with your material and measure. In this particular iteration of measuring enzyme catalysis, there are 2 experiments involved. The first of all is to measure the enthalpy of the turnover of your material, in this case, hydrolysis of this phospho-NPP. And you do this by having a relatively high concentration of enzyme and doing a long duration experiment. And what you're measuring is complete turnover of the injected substrate for the enzyme. You see here, you get some peaks initially, which correspond to the enthalpy. And then this decrease in heat you're getting here is presumably due to product inhibition of the enzyme. Once you've got this enthalpy value, you then use a very, very low concentration of enzyme, short duration injections, and you do a series of injections where you're increasing the concentration of the substrate. And effectively, these levels that you get after each injection are used to give you the rate of heat per unit time. And this can be converted into a rate of turnover using this enthalpy. You can see here that the values obtained by ITC are very close to values obtained by a conventional optical assay. So in the last 2 minutes, I just want to mention the other type of biocalorimetry, which is DSC. And in this, we have here in the LMB a DSC instrument, which uses robotic loading and operation. The active cell volume is 130 microliter, but we need about 360 microliter of a solution, about 0.2 to 1 mg per mil of our macromolecule that's going to undergo some sort of thermal phase transition. Again, it's using a lot more than the active cell volume, but that's because of the robotics needed to load and fill and clean the system. We have a wide temperature range and a wide range of scan rates. What we can get from a straightforward experiment, the data would look something like this for the unfolding of a protein. So we can get thermal stability, we can get a melting temperature under these 3 different pHs. We can get the enthalpy, which corresponds to the area under these excess heat curves, and the delta Cp, which is the difference between these baselines here of the native material and here of the unfolded material. Get that under a variety of conditions or for mutants or whatever. Here's just some examples of thermal shift, which you can also do using a DSC. So in this case, the ligand binds to the native state of the protein. Therefore, the higher the concentration of ligands, the higher is the melting temperature. So is increasing the concentration, we get an increasing TM. In this case, we're looking at cyclodextrin, and this tends to intercalate around aromatic groups, which are exposed in the unfolded state. So the ligand has a higher propensity for the unfolded state. So as we increase the concentration of the ligand, we destabilize the protein. And then just to show you here, something that I find quite interesting and is quite remarkable. In this case, somebody thought about looking at the DSC of human plasma. Human plasma contains about 400 proteins, I think. And so it's an incredibly complex mixture of solution. And what they found was that if they looked at plasma from so-called normal human patients, in this case 15, they obtained DSC thermograms, which were very complex in shape, but fairly reproducible. They found then that if they looked up in the literature, all the different proteins that are present in human plasma typically, and they normalized they measured those curves individually, then they normalized the curves relative to the typical concentration in a normal human, and they were able to then regenerate this profile fairly well, knowing what proteins are present, how much of them are there. But then what they then did was to look at some plasma from patients with a range of different diseases and cancers, and I think they did this somewhat randomly. Here's in the case of the blue trace, this is a patient with cervical cancer, and then the dark yellow trace here is somebody with Lyme's disease. What they argue in this paper and subsequently in other publications is that this doesn't represent a change in the levels of these proteins, but during disease processes, the very low levels of metabolites and products and other components associated with the disease, and these interact with the proteins present normally in the plasma and generate some sort of thermal shift, decrease in stability or increasing. And because it's a very complex profile, that means that the shape changes. Anyway, I suppose the real thing to say about that is calorimetry, both ITC and DSC, can be applied to quite unusual settings. Okay, here's the summary. So these are the take-home messages. Calorimetry is label-free and a very general method for any biomolecular equilibrium. And I should say it's not only about proteins. We can look at nucleic acids, lipids, bits of membrane, anything. For ITC, we are mainly looking at binding, and we get the association constant, stoichiometry, the enthalpy, and then the entropy is obtained by subtraction, the delta Cp if we want, and the change in protonation if we're interested. DSC is for thermal stability, so we get the Tm, the enthalpy, the delta Cp for the thermal phase transition. There's something called the Van Hoff enthalpy, which I haven't mentioned, which tells us something about the mechanism of unfolding, and again, changes in protonation. We can get changes in TM indicating ligand binding that we're going to hear about next week. And there are many other uses for both ITC and DSC, enzyme activity, the kinetics, where you're looking at broadening of the peaks, aggregation, and also there's a whole field of DSC used in formulation, particularly antibody formulation. But enthalpy and entropy can't be interpreted mechanistically in isolation because there are all these other contributions from solvation, dynamics, and so on. I haven't given any specific examples from work that we've done in the LMB, but here's just some papers that we've published over the years showing using both ITC experiments and also DSC. Here are a couple of reviews that I've written recently. This book is definitely in the library because I gave them a copy, one of the copies that the publisher gave me. So there's a chapter in there that I wrote on ITC. There's also this Nature Reviews Methods Primers, which came out last year. Yeah. And again, that's another review of ITC as a technique. These are the instruments that we have here in the building, and obviously for access, training, and discussions about how you might want to use them, then just contact the Biophysics Facility. That's it, and thank you.